And we are live.
Good afternoon, everyone. Thank you for joining this terrific session, exploring more in depth early age onset colorectal cancer regarding the causes of early onset colorectal cancer and how we can address the gaps that are still need to be addressed in depth. We're joined today by a distinguished panel, and I'll just begin by introducing myself, and then I'll ask each of our panelists to introduce themselves and their respective institutions. My name is Steve Gruber. I am at the City of Hope National Medical Center in Duarte, California. I'm director of our Center for Precision Medicine and Professor of Medical Oncology. Delighted to be here with you today to moderate this important and exciting session. This will be a 55-minute session with a vibrant discussion of distinguished panelists. And let me begin by asking Dr. Sanchez-Vega to introduce himself. Dr. Sanchez-Vega?
Hi, my name is Francisco Sanchez-Vega, and I'm an assistant attending computational oncologist at Memorial Sloan Kettering in the Department of Surgery and the Colorectal Cancer Service, and also in the Department of Epidemiology and Biostatistics. So yeah, in my case, I wanted to talk a little bit about the importance of being able to mine the electronic medical records and to integrate this information with information from large-scale genomic sequencing analysis. This is something that you've probably seen recently in some of the big genomic studies, starting with the TCGA projects a few years ago. Several projects that have been done in our institution have really mined the electronic medical record extensively, and right now we are moving in a direction where we are trying to stay away from manual curation and move towards automatic extraction of features from these electronic medical records. So yeah, if you've seen the talk that has been recorded, you'll see I'll discuss a little bit the case of the GENIE project from the AACR, and in particularly the GENIE BPC, the Biopharma Consortium, where a group of companies are collaborating with several institutions around the world in order to be able to extract clinical data and mine and do a complete annotation of all the clinical features, different lines of treatment, outcome for very large number of patients that will be shared with the public.
These are going to be really interesting topics to discuss, especially as we consider genotype-phenotype correlations and the importance of gathering accurate data for our early age onset patients. Thank you so much, Dr. Sanchez-Vega, for joining us today. My friend and colleague for many years, Dr. Pete Campbell. Pete, may I ask you to introduce yourself?
Yeah, sure, happy to. My name's Peter Campbell. I'm a scientific director and cancer epidemiologist, currently wrapping up, I guess, a 13-year career at the American Cancer Society before heading to the Northeast to be closer to Cindy, of course, among other advantages, I guess, to be at Einstein. My group primarily works in genetic and molecular epidemiology of colorectal cancer etiology and survival. And, you know, my main role and interest in being here today is as the principal investigator of the Colorectal Cancer Pooling Project, which is a large consortia study of about 26 or 27 prospective cohort studies from around the world, where we are in the process of pooling data from about 3.3 million people, including about 2,600 that have been diagnosed with early onset colorectal cancer. The overarching goal of, you know, investigating really etiology into a disease that we don't really understand too much just yet.
Dr. Campbell, thanks so much for that. And one of the things that we'll revisit when we come back to you for more in-depth thinking about causes of early onset colorectal cancer is the advantage of being able to use the types of prospective cohort studies that you've emphasized to look at risk factors that perhaps haven't been as fully explored as possible with that rich data set. So thank you for joining us. Dr. Kimi Eng, who was scheduled to be joining us today from Harvard, will unfortunately not be able to join us today as she is on service. But I would like to turn our attention to introduce Dr. Cynthia Sears from Johns Hopkins. Welcome, Dr. Sears.
Oh, thank you very much. It's a pleasure to be here, and good afternoon, everyone. I'm Cindy Sears. I'm at Johns Hopkins, as just stated, and I'm an infectious diseases specialist, so interested in how the microbiome and particular bacteria may be influencing colorectal cancer in general, but now beginning to pivot to try to understand this better in early onset CRC, where there actually are no dedicated studies as yet. So lots of great questions that perhaps we'll get into, but it's really a pleasure to be here. Thank you.
It's an open book, and we look forward to having you show us how to write that book. So we'll be back to you in just a few moments. Thank you, Dr. Sears. Let me now introduce and welcome Dr. Caitlin Murphy from UT Southwestern.
Well, good morning or good afternoon, depending on where you are in the world. I'm Caitlin Murphy. I'm a cancer epidemiologist at UT Southwestern Medical Center in Dallas. A lot of my work is around early life periods, so these critical periods of growth and development like infancy, gestation, and childhood, and how things that happen during those times may influence the risk of colorectal cancer many decades later. So looking forward to talking with you all more about that today.
Thank you so much. And when we come back to revisit some of the hypotheses that you were exploring in more depth, I'll just begin by sharing one of the pearls that was described to me early in my training by my friend and colleague, Dr. Stan Hamilton, who formerly was at MD Anderson and now is the chair of pathology at City of Hope. And he said, Steve, what you have to remember is colon cancer is a pediatric disease. It takes decades to develop, and we need to understand early life exposure. So I've always kept that in mind, and it sounds like you'll be able to guide us in our thinking even more with respect to Stan's early thinking. So let me now ask Dr. Jose Pereira to introduce himself from Madrid, Spain.
Yes, thank you, Stephen. It's a pleasure to be here. Glad to be invited. So I'm Jose Pereira, I'm colorectal surgeon here in Madrid, Spain, from Fundación Jiménez Díaz Hospital. I have been working since more than 10 years, focused on early onset colorectal cancer. Firstly interested in genetics, but now I'm coordinating two prospective study. One in Spain that is a multi-omics study that try to see the interactions between not only genetics, epigenetics, but also exposome and microbiome. And then starting regarding disparities that are arising now from early onset colorectal cancer, we are now building a consortium, an international consortium worldwide with more than 20 countries around the world with in 5 continents in order to see or to explore the differences between the geographical areas about early onset colorectal cancer.
Thank you so much. It's great to have you here, and your work known to us, and we're looking forward to learning more about your multi-omic studies, especially as we think about the way in which you're integrating the exposome more broadly than perhaps we've considered before. So welcome. Great to have you and your expertise here. Let me finally welcome Dr. Cavastro. Dr. Julia Martina Cavastro will be speaking to us about diet. Please introduce yourself.
Good evening. My name is Giulia Martina Cavestro, and I am an associate professor of gastroenterology here in Milan. And I am the coordinator of a multidisciplinary group for the hereditary tumors here in Italy, and also for early onset colorectal cancer. Well, this evening, I will present to you, I would like to share with you our project, our Demetra project, on the association of specific dietary and lifestyle factors with early onset colorectal cancer.
Thank you so much. It's great to have you here. We look forward to your comments. And let me invite the panel to really bring forward questions and make this as interactive as possible as we're working with each of you to talk about your individual areas of expertise. For those that are participating live, we welcome to have your comments in the chat. That will also help guide our questions and responses to you to make sure that this is a vibrant and meaningful discussion. Let me begin by asking the question to you, Dr. Sanchez Vega, about the way in which tools like artificial intelligence, machine learning, and augmented intelligence can really help provide us with insights that will lead us to new understanding of risk factors, especially since, at least as I tried to summarize yesterday in a talk that describes where we are, there's more that we don't know than what we do. So how are you bringing your machine learning approaches to help provide insights?
Yes, I would in particularly highlight two questions that these methods can help us with. One of them is scaling up the number of patients in each study to orders of magnitude larger than what we were doing before. As I said, when the TCGA started doing some of their projects, the first TCGA paper about colorectal that was published in Nature in 2012 contained around 270 patients. Some of the more recent manuscripts that we've submitted from MSK this year are already in the order of 2,000. And some initiatives like Genie are now working with 100,000 patients across different cancer types, but this will continue to grow. So of course, if we don't have robust statistical tools and machine learning approaches that allow us to interpret and to process all this data, we won't really be able to scale up. Another important aspect is the need to do multi-dimensional integration. So I believe that the field is moving towards some new models where we combine features extracted from radiology images with clinical data extracted from the electronic medical records, or with information from the microbiome. We are now, for example, starting to work a lot with liquid biopsies as a non-invasive approach for longitudinal monitoring of the patients. And of course, we are also trying to integrate this with genomics, and these genomics become more and more complex. So some of our projects are using targeted panels that only interrogate a small number of genes, but then it's very common to work also with whole exome data. And more recently, we are also moving towards whole genomes. So all this creates really incredible challenges. Even just dealing with that sheer amount of data, when you start working with whole genomes for 1,000 patients, this is huge. And then you bring in the MRIs and you bring in all these other data modalities that I mentioned. So that I think that there are very, very important challenges ahead. But it's very promising and it's very exciting.
Let me ask you, if I may, to follow up on the way in which you're using artificial intelligence or natural language processing for data abstraction from charts. How much abstraction still needs to be done with manual curation? How do you balance what's needed by expert abstracting curators versus what you can accomplish with NLP, for example?
That is a great question. So I can give you an example with some of the current projects that we are working on. We have at MSK, we have this platform called the MSK Impact Clinical Platform, where many of the patients that arrive at MSK get their tumor sequenced, and then their information goes to a central database that can be accessed by researchers. So yes, when Impact is run and there is a report generated for the patient, we always know if the sample that was sequenced was a primary tumor or if it was a metastatic sample, a metastasis. And if it was a metastasis, we know which organ was sequenced. So we know if it was a liver met, a lung met. So that is really good as a starting point. But in some projects, we really wanted to be able to know all the sites of mets for a given patient. So, some of the early strategies that we've been using is, what if we go to the electronic medical record and we start mining the ICD billing codes to see which sites of metastasis were billed for each patient. And that is a good starting point, but is really far from perfect. So the good news is that by querying these structured databases that already exist, like the database of ICD billing codes, we can recover information for thousands of patients very quickly. Now, if you really want to go one step further, you could have somebody that go manually reviews each of those EMRs and see if there was really a metastasis here or there, or if some of them were missed by the billing codes. And thinking of it in more towards the future, we could also think of using NLP, natural language processing techniques, to mine the pathology reports and sometimes write the impressions of the surgeons when they took the sample. We could also think of automatic feature extraction from images, from medical images, that you do some kind of image, and it automatically identifies all the sites of MET for each patient, and that goes into a centralized database. So we are able to get some of this information now, but there's a lot of room for improvement, and there's a lot of research going on to be able to turn all these, for example, all these unstructured tests that sometimes is in form of PDFs and written reports from the doctor, to turn it into a structured text, so that can be very easily queried by computers, and we can do statistics on.
So Dr. Perea, thanks for outlining the way in which you use both structured and unstructured data to really characterize these patients. But let me ask you in more depth with respect to early age onset colorectal cancer. Where are the key areas that you are trying to explore, given the really large data sets that you're now able to understand and analyze?
Yeah, I mean, the limit. Yeah, the limit is on your imagination. So there was this very recent manuscript that was submitted and is currently under revision by Dr. Andrea Sersek and Sophia Stadler, where they were looking at early onset colorectal cancer. And for example, in that case, they were able to extract from this EMR all the symptoms at presentation. And because we had this data for a set of around 700 early onset patients and 700 average onset patients, you can start comparing, as I said, symptoms at presentation with the genomics of each patient. You can start thinking of correlating that with other aspects, clinical pathological features like MSI or MSS, left-sided versus right-sided versus rectal. There's really very, very interesting questions that can be addressed, and unfortunately, I think that it's very difficult to get information outside of that EMR that maybe in the future may also become available, such as lifestyle, whether the patients do exercise frequently, or what kind of medications they get. Did they get a lot of antibiotics in recent years or not? There is a lot of information that is still outside of the EMR, and that maybe in the future might be accessible.
We'll come back to that very important topic in just a moment, but let me turn it over to Pete Campbell to think more about risk factors, because what you've just described, and I love the work that Sophia Stadler and others have done. I think it's exciting to be able to look at symptom presentation and correlating that with genomics. One of the advantages of prospective cohort studies is that you get to define what the exposures are in advance, Pete. So could you just describe the depth of the information that's available across the pooled studies that you're using, and what are the exposures that are available, and what's missing?
That's a great question. So what's available is, so maybe I should back up. I suspect not everybody listening is a PhD trained cancer epidemiologist. So when Steve talks about a prospective study, we're talking about studies like Nurses Health Study, Physicians Health Study. These are the studies that you hear about on the news all the time. So we've assembled 26 different prospective cohorts that have agreed to participate. And with each of them, they generally began in the 1990s, so they've had a lot of follow-up time until, usually about middle, 2010, 2015 or so. So we take those 26 different studies, and as Steve was mentioning, like we have to harmonize. We have to make studies that were done in studies done in China, studies done in Australia, studies done in the United States have comparable data. So what we tend to have is, it's almost like a lowest common denominator. So we will have some diet data, like we'll have Western diet patterns from factor analysis. We may have red meat intake, fruit and vegetable intake. We'll have body mass index, so height and body weight. We'll have some data on physical activity, but that's notoriously difficult to harmonize across multiple studies. We'll have aspirin intake. So if you think of like almost all of the usual suspected risk factors for any given cancer type, we will have that data in spades, a lot of data on that. Some of the data that will be more sparse that we would really love to have more of would be, maybe, breastfeeding practices, early life antibiotic exposure. Some of that'll be, not uniformly available, we expect. But maybe most interestingly, what we will have is pre-diagnostic blood samples. So these were bloods usually 7 to 10 years prior to diagnosis. So these were truly prospectively collected. And within those blood samples, we can detect, or at least we hope we will be able to detect biomarkers for risk. And the biomarkers that we've chosen to focus on are those that we suspect are affected by the lifestyle changes that have happened since the 1960s, 1970s, when we know, like my age group, where we're seeing some increasing trend, we're trying to focus on those. So those biomarkers will be reflective of gut dysbiosis, metabolic dysregulation, so like diabetes-associated markers, as well as a host of markers related to systemic inflammation. So we think we have like a good mix of hypothesis testing and some exploratory work in, it's a range, I think it's 110 different biomarkers that we've selected. But we applied for an NIH grant back in November. It scored very well. I'm not sure that'll be funded on the first application, but hopefully with revision, we'll be able to get it funded the second time.
So let me ask you to elaborate a little bit about exploratory analyses versus hypothesis-driven ways to approach early age onset colorectal cancer. Because Pete, what you described in your introductory remarks about having 2,600 patients identified from your 3.3 million people overall is really a remarkable resource to be able to answer questions. Let me just ask you to think about the exploratory analyses before addressing some hypothesis-driven components.
Sure. So the exploratory, I suspect, or I suggest that in some sense, almost all of this is exploratory. So we're coming into this with the assumption that the risk factors for earlier onset cancer outside of the familial syndromes, they probably look similar to older onset disease. And that's based on, generally smaller studies of 100, maybe 200 early onset cancer case or colorectal cancer cases, sometimes including adenomas. So we know that things like BMI, diabetes, lack of aspirin use, maybe physical inactivity, they appear to be associated and relevant to early onset colorectal cancer too. Again, I don't think anything is definitive yet. I think that's one thing we've heard repeatedly over the weekend, that these are smaller studies, but they're still pointing in the same direction. And Steve, as you know from GECKO and Correct in the colon CFR, I don't think the paper's published yet, but we've also shown that the molecular characterization of these tumors looks a lot like older onset disease does too. So I don't think we're looking at a brand new disease entity. We're just looking perhaps at a disease that happens earlier in life because of exposures that have changed much earlier in life, and for people that were born in the 70s and perhaps the 1960s as well. In terms of, you were asking about exploratory, we're using a proteomics platform to measure 92 proteins associated with, or potentially associated with, early onset disease. If budgets were limitless, I mean, we would measure thousands, tens of thousands of potential proteins through, other proteomics platforms or metabolomics platforms. But budgets being what they are, we chose to focus on what we think is the most fundable, which is a little bit more hypothesis testing than exploratory.
Pete, I'm going to ask you to elaborate on one specific component of biomarkers as it's related to the rapidly emerging field of liquid biopsy. And before I ask you to answer that, I just want to point out that in just a moment, we'll be switching things over to Dr. Sears, where I'm going to ask her to comment more specifically about infectious diseases. And maybe you could set the stage for her by talking about what those resources of collected biospecimens would allow you to do, not only for things like liquid biopsy, genomic characterization, but how about things like antibody tests to look for changing profiles of the microbiome and infectious disease exposures over time?
With funding, we'd be able to test just about anything. There are caveats to that. So one reason that we selected the series of panels that we chose for our first project in C2P2 was so that we would not burn up too much sample. So for each cohort that is, often contributing maybe 20 to 50 early onset case samples, these samples are invaluable. To have 1,000 or so blood samples from people prior to the diagnosis of a cancer, they're truly invaluable. But they're also very limited in terms of the blood volume that has been allocated or allocated. So with our first series, we wanted to show that we could learn something new about the etiology of early onset colorectal cancer while not burning up too much sample. It sounds much less sexy, if you will. But there are very real limitations to what we can do with other people's sample. Metabolomics, the cell-free DNA tests, Steve, that you mentioned, I mean, they burn up a ton of serum at present, at least the ones that I'm familiar with. So there will be a time and place, I'm sure, when we can talk our collaborators into allowing us a little bit more sample, but I'm afraid we're not quite there just yet. I think as those technologies get better and require less serum plasma, we'll no doubt have an easier time talking them into those platforms.
We'll come back to you later because I want you to help us at the end think about what are the next steps that we should be thinking about. What samples do we need to collect for the next generation of studies?
Sure.
But I'll come back to you about that because I really want to invite Dr. Sears to share her expertise with infectious diseases to think about this really troubling problem of early onset colorectal cancers.
Well, thank you very much, Dr. Gruber. I think in our world, if you will, of the microbiome, we're looking at how the microbiome may be influencing colon cancer almost regardless of age, based on whether there is specific bacteria there or particular communities. And the long-range vision is our hope that we can contribute to a diagnostic approach or a biomarker approach to allow us to identify these younger individuals. And at some level, I believe that things like hereditary colon cancer, such as familial adenomatous polyposis and EAO CRC will actually help us with, if you will, sporadic CRC in the older individual. I just, as was just said by Peter, I think it's a change in timeline, not necessarily a new disease. And when you look at the history of exposures, we certainly see concerns such as the obesity epidemic, the emergence of Clostridium difficile. I will mention, that was not a disease in children when I trained, but it became a disease in children that is quite established now. That also coincided with the increased emergence of EAO CRC. So I think the epidemiology and history is clear. So from our perspective, we hope to go back in time. So at Hopkins, we have availability of pathology back to the 1970s. And so we hope to look from there to now and then prospectively in new patients at the possibility that there are biofilms, which are these really dense communities of bacteria that form on the surface of the colon and invade the mucus, and which unequivocally change the function of the colonic epithelial cells, in which we've been able to show are pro-carcinogenic in some people. They're incredibly common in sporadic colon cancer. Over half of colon cancer has them, and in some very preliminary work, we're seeing a similar detection rate in younger individuals with colon cancer. And at the same time, we hope to look at the epidemiology of some of these bacteria that have been demonstrated to be pro-carcinogenic in mouse models, and also epidemiologically linked to sporadic colon cancer. We know that some of those organisms are commonly colonizing children, like the polyketide synthase Escherichia coli or PKS coli are very common in children, but there's no disease. These are just common in the community. At DDW, we will show that there's about a third of a colonoscopy population that is colonized with these particular bugs. Now, similar to H. pylori, I doubt they're all causing cancer, but you know, the trick is figuring out who's at risk. And I think all these exposures people are talking about is so critical to integrate into these data.
Well, I'm so glad you brought up the example of H. pylori, because obviously that Nobel Prize winning work changed the field for thinking about gastric cancer. And I wonder, where do you see our understanding of the microbiome in colon cancer with respect to, say, for example, what we know about gastric cancer at this stage of our scientific development?
Well, as everyone will recall, you know, H. pylori was declared a class 1 carcinogen, I think I got the wording right, by WHO in like 1994. So pretty early in the evolution. And that was based on both serology, which you've been discussing, sort of looking back at exposure and coming to understand that if you got rid of the bug, you had less of the disease in a relative, and dysplasia in a relatively short period of time. In truth, over a couple of years, and that data has only been reinforced, including in a recent paper in the New England Journal. So, you know, it's very, very valuable data. So as has been discussed, I do think trying to look at microbial exposures using serology, and one approach we're going to try is something called FIPSeq, and this is it's a high throughput approach that can be used to look at antibody responses to a broad spectrum of antigens that was developed by Ben Larman in our pathology department at Hopkins, and has been applied in different disease settings. I might mention a paper in Science with looking at measles. You know, we all know measles makes kids incredibly sick. And then they have, after measles, ongoing mortality and ongoing excess morbidity, including diarrheal disease. Well, this paper using this approach was able to show that infection with measles blunted the antibody response over a broad spectrum of antigens. So we're hoping to work with Ben and dial into his assays some of these critical bacteria that are, it's just not the common ones you've heard, but there's some others emerging that we would try to get into the screen to try to look at exposure as one way to get some clues to why this may have emerged.
So let me ask you to follow up on that by thinking about the impact of antibiotics. And specifically, I once heard a lecture where an infectious disease and microbiome specialist described antibiotics as the atomic bomb of bacteria in the gut flora. And of course, the gut reconstitutes its flora. What's the short-term and long-term impact of antibiotics on microbial flora that might impact colorectal cancer risk?
Yeah, it's a great question, and there is a variety of epidemiologic data that look at impact of antibiotics on a variety of cancers, but colorectal cancer. We had the opportunity to do a study using the UK database, which is a very large primary care database, the CRPD. Looking at 28,000 cases of colorectal cancer and about 137,000 controls. And what was fascinating is that the biggest impact of antibiotic exposure was in the right colon. And there was no impact on left-sided colon cancer by antibiotic exposure, but there was an impact on rectal cancer, and in fact, it was the inverse. So there was an increase in right-sided colon cancer and a decrease and rectal cancer. And we were able to dial it down because of the size of the data to a couple antibiotic classes. Now, why might that make sense? Well, after thinking about it long and hard, and this focused on oral antibiotics, if you think about it, we don't absorb all of any antibiotic we take. So there's spillover into that right colon. And my hypothesis would be that you do detonate the population there, and particularly the mucosal bacteria, which I think are potentially the most important to pathogenesis. And we have also learned through studies from David Relman and others that, we think the microbiome reconstitutes itself eventually with antibiotics, but there are some long-lived effects. And particularly, I wonder if these occur at the mucosal surface. And so you get long-term change in the colonization and potential impact. So in my mind, that's what makes sense. And the last thing I'll mention there is it's not antibiotics just before disease. There appears to be no confounding by indication, but rather the exposure had to occur more than 10 years prior to be linked to an increased risk, which I thought was almost remarkable, given that we think of a decade as the minimum time for emergence of clonal expansion and being able for the gastroenterologist to be able to see a tumor in the gut.
That is a beautiful segue to having Dr. Caitlin Murphy about early life exposures. And I would just say I'm fascinated and really surprised to learn of that right-sided, left-sided difference. But the hypothesis that you're putting forward is really intriguing to consider. It also suggests that Dr. Sanchez-Vega is going to be employed for a long time for the types of large data set analyses that will be required. But Dr. Murphy, can you describe for us how you are thinking about early life exposures?
Yeah, I think it's important to first maybe take a step back and talk about why we should study early life exposures. And we talk a lot about this birth cohort effect and generational changes in the 1960s and 70s. And those are fun observations to describe for epidemiologists like me, but they actually have a lot of meaning because they point us to early life as the period that really matters most. So I think the most classic example of a birth cohort effect is what we see in lung cancer, where we know smoking behaviors change dramatically with generations, and we see those same changes occur in lung cancer incidence and mortality rates. So I've been really trying to dig deep into what early life factors might be contributing to the increasing incidence rates that we're observing of early onset colorectal cancer. You can imagine, though, that sometimes it can be tricky to study early life exposures. I see a couple of questions in the chat about, starting new data collection. Well, if we wanted to do that, it would take years and years and tons of money to collect information from infancy to adulthood. And quite frankly, many of us might not be here by the time that data collection was finished. The other challenge is, asking people about these early life exposures. It's really hard for people to remember, oh yeah, I think I took X amount of antibiotics when I was an infant. I mean, that's impossible kind of detail to really recall. So I've tried to get creative, and I have found this pregnancy cohort. It's called the Child Health and Development Studies. It was a population-based cohort of about 14,000 women who were receiving prenatal care in the 1950s and 60s, so that exact time period that we're interested in the Kaiser Health System in Oakland, California. Now, its original purpose was really just to try to understand pregnancy-related outcomes, but they have followed this particular cohort over time and can link it to the California Cancer Registry to identify cases of cancer that have been diagnosed in the offspring. And this is quite an amazing feat because we've essentially followed those offspring for 50 and 60 years now, and so can really establish sort of patterns and relationships between what happened in early life and gestation in their childhood and the risk of cancer many decades later. And to give you a high-level overview, some of what we're finding so far is that in utero exposure to certain medications, so for example, there's an anti-nauseant called Bendectin that was quite popular in the 1960s that seems to play some role. A synthetic progestogen that was used to prevent miscarriage, much like the drug diethylstilbestrol or DES, except this was a progestin and not an estrogen. And then sulfonamide antibiotics. So we didn't see an association with tetracycline or penicillin. And again, this is in utero exposure, so there could be a different relationship later in life. But it was the sulfa drugs that seemed to really increase the risk of colorectal cancer, maybe because we know that they do cross the placenta, so something having to do with that. Really interesting things that we're seeing, and can continue to work with this particular cohort to try and understand, what was happening during those critical periods of growth and development that could explain what we're seeing today 50 years later. Oh, I think you may be on mute.
Sorry, just want to make sure there was no background noise there. But really an extraordinary prospective cohort study that allows you to look at those not only early life exposures, but prenatal exposures as well. And especially the work that's been done with respect to thinking about prenatal exposures for leukemia. It's such a promising area for exploration. Let me ask you to turn your attention perhaps a little bit later in embryologic development, but early life exposures, pediatric exposures. Are there ways in which you can look at those data over the course of time that gets us away from some of the data where we're just looking at trends that are vulnerable to ecologic fallacy? And just to frame this for a larger audience, it's really easy to come up with hypotheses to say, well, this risk factor changed over time, and so did this disease incidence. But that doesn't actually mean that there is a causal relationship. But the type of data that you are describing, Dr. Murphy, really gives the ability to look specifically at individual risk factors in individual patients. What are the strengths and limitations of the data that you're able to explore for postnatal early life exposures?
Yeah, so I should clarify that we have information of the first 5 years of childhood, which we have not begun to explore yet. So there is opportunity to look at childhood use of antibiotics, childhood obesity, other growth-related trajectories, and how that might be related to the risk of colorectal cancer in adulthood. We're just not there yet, but do have those data available. One of the challenges is that we have a gap in our sort of timeline where we don't have complete information on everyone during sort of young adulthood, adolescence, and mid-adulthood. So we don't know for sure sometimes whether the association we observe may be due to its relationship to some other risk factor or exposure in adulthood. So for example, we can see that maternal obesity, having a mother who was obese before she was pregnant, is associated with risk in the offspring. But that may be because they shared some kind of environment after the child was born that is also related to the risk of colorectal cancer. We just can't quite tease that apart. So that is one of the limitations. But I think certainly the strengths of having the prospective follow-up and the size of that particular sample far outweigh some of those other limitations.
Pete, let me come back to you for a moment, if I may, just to comment and reflect on the differences in the cohorts that you're studying, where we have data, say, from the physician's health study, the nurse's health study, where there are exposure data, for example, physical activity in early adulthood compared to the data that Dr. Murphy has available. What's the limit of resolution that you have?
We can definitely fill in some of those gaps. Recalled physical activity is a little difficult. I think the best we can do there is what some of the earlier Harvard studies did. I'm not going to remember which one it was specifically, but they asked about sport participation. So we can learn about what the participants in the study did in, say, college. So Caitlin's got the earlier part of life covered, then some of these Other studies will fill in, roughly ages 20, 30, and beyond. Some of that will be recalled. Generally in prospective cohort studies, we enroll beginning at about usually no younger than age 35. And that's something that I think we really should reconsider in the future. It would require, a newly funded prospective cohort of younger people, say beginning at age 18, 18 or 19, when they can properly get consent and informed consent.
And Dr. Murphy, let me come back to you, because the work that you're doing is incredibly exciting to be able to explore those really early embryonic exposures. What are the questions that you find most exciting and interesting to answer right now with respect to colorectal cancer?
I think someone said it well earlier. In some sense, everything that we're doing is exploratory. And the more that I work and dig into this cohort, the more I am just personally convinced of the importance of early life. And so really trying to understand what factors those were, perhaps what factors may come later in life that mediate that effect, and how we can make an impact today based on something that occurred maybe in the 1960s. So, for example, a lot of the drugs that offspring were exposed to in utero may not necessarily be used today as drugs, but are persistent in the environment, for example. So really trying to think, how can we translate some of these findings about gestation and infancy and in utero exposures to making a difference today? And it seems to me that a lot of those signs point to the environment as sort of the next step of things to do.
And what I'd like to do next, Dr. Murphy, is ask you to comment about the richness of your data set, or the limitations of that data set, to be able to answer questions about disparities. In the California cohort, it looks like that you're studying, it's certainly a diverse population, all had insurance. And as we look at some of the real challenges with early onset colorectal cancer right now, it is not a burden that's shared equally among all populations. What's the breadth and depth of your ability to look at disparities within your prospective cohort from the 1950s?
Right. As you mentioned, the cohort is very racially and ethnically diverse. About 1/3 of the offspring, of which there are maybe about 19,000 offspring, are racial and ethnic minorities. And thinking about the fact that this cohort was started in the 1950s and 60s, I think is really impressive that we do have such racial and ethnic diversity. It's also pretty socioeconomically diverse, so the median income is about split among people, 1/3 over, 1/3 at the median, and 1/3 below the median. So you do see some diversity there as well. You pointed out that most people had insurance. I just want to point out that that is at the time that they were born. So we really don't know what their insurance coverage has, occurred over the rest of their life, but certainly have done some follow-up studies of the cohort to try and get at that information that we're kind of missing in that young adulthood and present day period. So I think there's a lot of opportunity to try to understand differences in the exposure experiences by race, as well as how those differences may impact differences in disease risk.
Well, we certainly look forward to learning more as you explore that extraordinary cohort. And let's turn our attention to Dr. Perea, because you've really made some exceptional contributions to our understanding of colorectal cancer, and especially within the context of early onset. But how are you bringing your focus on disparities in Spain and thinking about the multi-exposome approach to looking more broadly for causes of early onset colorectal cancer in your studies, Dr. Perea?
So we have to separate two main studies. Firstly, the Spanish cohort, because it is very uniform regarding races, and then from that point of view, firstly we started to analyze molecular approaches, and of course we saw that compared with late onset colorectal cancer, there were a lot of differences. So from that point, we started to see that there were a lot of correlations with inflammation and other pathways related with metabolism. So from that point, we started to integrate another points, not only the molecular, because in my opinion, I think early onset colorectal cancer should be the best integrating system for all the factors that has been already told. For example, exposure, for example, the microbiome. So from that point, nowadays we are analyzing all the cord from different point of views. We collect from them a stool, we collect blood samples, plasma samples, and then we plan to make and integrate a study for multi-omics approaches just to see what's going on, because you have to take on account that, for example, the Spanish population is completely different from the United States. For example, here in Spain, we don't have too much obesity, more or less. I mean, because nowadays maybe the last 10 years, 5 years, obesity is increasing. But for example, for people around 40s and 50s, there is not so much obesity compared with the United States. So the first approach is just to explore what's going on about carbohydrates metabolism in order to see what's the role in our population. That is, for example, the multi-omics approaches here. And then another point is disparities, because we saw that, for example, if you see the world map, the incidence is increasing about early onset colorectal cancer, but that is not uniform from different countries. And we ask ourselves, why is this? Because there should be a lot of factors related with that. So not only geographically, but also if you take on account, you have already told races. For example, there are differences regarding races. For example, for survival, for example, I have collaborated with a study presented with Andreane Holowatig now in the American Association Cancer Research meeting, and we showed that molecularly the races are completely different if we focus on early onset colorectal cancer. So all of these we have put in a bag. And then we have planned, we are now developing the study that is GeoCode in order to see what's going on about early onset colorectal cancer all around the world. That is the idea.
I'd like to elaborate a little bit on the way in which colon cancer screening is done in Spain, because one of the hypotheses that's been explored is that differences in screening might actually lead to differences in detection. And certainly countries have made strategic public health choices about how to best approach screening for colorectal cancer. How is screening in Spain conducted, and what are the impacts on the way in which you are exploring studying colorectal cancer in early onset more specifically?
So it is an interesting question, because now we have sent a manuscript for publication, making in the same direction as Heather Hampel presented in Gastro about what is the proportion of early onset colorectal cancer that is preventable. She showed that it was more or less than 16% regarding familial components, and with our data, it is compounded with a lot of centers around Europe. And the preventable proportion of early onset with our data is only 5%. So that is one point, for example, and just to see the differences between areas. And then another point, for example, in Spain, we have a screening system, but starting at 50 years old. If you don't have any hereditary form, or for example, if you don't have familial colorectal cancer component. The problem is that here in Spain, we don't know what's going on about early onset colorectal cancer regarding incidence. So for us, it's so important to find markers, just to develop screening strategies, just to define in risk subpopulation, just to start with them specifically, the screening maybe in earlier ages.
So you know this field quite well, and I'm just going to ask you to project without data, I just am interested in your thinking about the future. So looking into your crystal ball, where do you think liquid biopsy is going to play a role more broadly from a global perspective of being able to identify early onset colorectal cancer? And specifically in Spain, is that something that you see that liquid biopsy might be playing a role?
Of course, that is in the same direction that I have told. Not having so much screening for people younger than 50 nowadays, we should find specifically markers for early onset colorectal cancer, and if we find some of them, of course, liquid biopsy should be the first approach, just to keep in mind.
Well, it'll be interesting to see where this goes, especially when we think about the resources of the serum and plasma banks that have been collected so beautifully by those people leading our cohort studies over time. We'll come back to you more to think about international consortium studies, Dr. Perea. But let me turn to you, Dr. Cavestro, to think more about diet. How are you approaching studies that will give us insights into dietary contributions to early age onset colorectal cancer?
So our study, we know that early onset colorectal cancer is increasing globally. On the other hand, we also know that latest finding based on Italian cancer records covering almost 36 million Italian residents, that is the 60% of the population, told us that in Italy there is a drop in early onset colorectal cancer. So we are in a special position, and we can be the real control group. So indeed, we cannot have, it is hard to find biologically consistent interpretation for this finding, as we said, and we lack evidence to support the claims of ethnicity factors or protective microbiota right now. What we know is that indeed we have the Mediterranean diet, and here in Italy is maybe the most frequent and most important dietary factor that we have. So in our mind, we set this study, and today, right now, 30 centers around the world are participating in this study, and we, the Italian cohort could be the control group indeed, and we are 13 centers, and there are some other centers around the world, from 8 centers from USA, from Norway, Finland, Spain too, and Germany, so all around the Europe. And we aim to have a single shared semi-quantitative questionnaire in order to have the same question, the same questionnaire. And Mr. Johnson asked us if it is possible to have a sort of questionnaire or something else to explore early habits, young habits. Well, I think it is quite tough because this questionnaire is 35 pages. It is an online questionnaire, and it will be quite tough, I think, to fill. But we have very young patients, so very motivated patients, and they will give us the right question. I think giving this type of questionnaire to the mother or to healthy persons, I'm not sure they will feel appropriately the follow. Anyway, we also explore and we also explore smoking habits. As Jose told, we don't have so much obesity, indeed, smoking habit, and we are going to try to understand usually habits of these patients in comparison to normal controls.
And the fact that diet changes over the course of a lifetime is also a particular challenge to think about the exposure to a diet at any given interval of risk. But as we're thinking about early onset colorectal cancer, at least we have the advantage that it's a compressed time frame where you have the ability to actually measure those risk factors. So if using your experience for using semi-quantitative food frequency questionnaires to really quantify dietary exposures, what would you say would be the best intervals to assess diet for those who might be at risk for early onset?
So we ask for the diet 2 years before the diagnosis to those patients, because we think it's quite tough to realize and to remember the diet before 2 years, the diagnosis. And after that, we are going to ask every year for the diet. It is interesting to say that indeed those kind of patients usually change their diet after the diagnosis, also in Italy. I mean, so I think we are going to have some information, but I mean, they tend to change their diet. So.
Well, let me open it up to the group, because this has been an absolutely stimulating discussion covering high dimensional data and informatics, infectious diseases, epidemiology, early life exposures, multi-omic exposures, diet. And if I could just ask each of you, and perhaps just raise your hand if you'd like me to invite you to comment specifically. We need to invest research dollars from foundations, from government support, to be able to address these types of questions. If you were to propose a provocative question based on your expertise to say, where should we be investing money right now to make sure that we are better understanding early onset colorectal cancer, and you're the one writing the check, where would you put your resources to be able to make sure we are investing in understanding risk factors for this? And please feel free to advocate for your own area of expertise. But where should we be investing our funding? Dr. Sears, let me begin with you.
Thank you. I think my answer would be, this is not a rare disease anymore, but at each center, there's relatively small number of cases. So if I could dream, we would create a network that is of communication and a way to contribute, maybe similar to the TCGA, a big data set and some parallel sample collection that would allow, hopefully, the exposure questions to really be pursued. And of course, I'll put in a plug for the microbiome, but I do think it's the epidemiology, exposures, and a sufficient population that we need, and it's hard to get it at one institution.
Dr. Sanchez-Vega, let me ask you to comment on that, because that really reflects a lot of what you were talking about with Gini and impact and the strength of really large data sets.
Yeah, I agree, and I think it's a great point. And I would stress also that it's very important not to just connect samples, to make bigger sample sizes, but also connect experts, because we won't make progress if you have a group of experts on the microbiome and a group of experts on epidemiology and genomics. You need to bring all these people together. which in a way was one of the big things also with the TCGA. They were combining clinicians and bioinformaticians, and so yeah, I think it would be a great way to use funding to connect experts and build expertise and integrate all these different aspects.
I think we need a U award or a P award type of mechanism where, the best investigators in the world put forward their best ideas on what is leading to this, these troubling trends, and NCI or another, very high dollar funder gets to select among those best studies which are most complementary and most likely to really understand and prevent this disease in the future.
Well, we are really right on time here with respect to, I think, a major theme, which is collaboration of the very best thinkers in the field with large data sets that get to help us answer questions, some of the questions we haven't even thought of yet. This has been just a wonderful opportunity to learn about your expertise and visions for the future. I want to thank each of you for joining, and as we've observed the comments on the side, hopefully we've been able to address some of the comments more specifically, but we're really grateful for this opportunity, and I want to thank each and every one of you for being with us today. Thank you.
Well, with that, I want to also join in, and I want to thank each and every one of you. I had a feeling this would be a fantastic panel discussion, and you did not disappoint at all. Our theme here at the foundation is collaboration, and I have a feeling that we'll continue that. I hope to have all of you back on at some point this year to further this discussion. In the meantime, have a great afternoon, and we will see you at some point in the future.
Thank you.
Thank you.
Thank you very much.
Thank you, everyone.
Thank you.
Bye-bye.